Welcome to another session on Botany Insider, a free session which will help you to practice out the previous year questions in the best manner possible. So, in this particular session, we'll be dealing with the previous year questions and we'll be dealing with various other segments as well. If you're new to Botany Insider, please make sure to hit the subscribe button. In addition, to download our application from the iOS as well as from the Play Store. If you're new to Botany Insider, these are the main mandatory things which you can do and get connected with us in the best manner possible, right? Let's begin. Before starting, we are having a number of live courses ongoing on our application. So we have a pre-YQ practice course for the June 2024 batch students. We have our examination in 2 weeks, exact 2 weeks from today. It's 10th. So you have your examination from 25th to 27th of June. So most probably the examination will be on 25th itself, because every time in every cycle of examination, the life sciences is the first exam to be conducted, because maximum number of students apply for the life sciences stream, right? So if you're preparing in the best manner possible, have confidence in yourself, have some patience, and just give your best shot in the upcoming examination, right? We have a number of other courses as well: full course, crash course, PYQ course. We have mock test series also available. We have hard copy notes also available. So those of you who are willing to enroll in any of these things, you can download our application and get yourself enrolled. In addition, if you're planning out for the December 2024 examination, if any one of you who is planning to get enrolled in the December 2024 course, so our classes for the same will start very soon. You will get live and interactive session, all the 13 units, part A, B, C, everything will be dealt. Hard copy notes will be provided to your doorstep. All the recordings will be available with no constraint of utilization. You can use the videos as many number of times. Daily practice questions will be discussed in the class. Previous year question 10 years will be solved. Regular doubt sessions will be there. Weekly test and 2 revision cycles of the entire sub syllabus will be completed, right? So this was about the Botany Insider and the work that we provide and offer to our students. This is the very first question for you all. Phosphatidyl inositol is unusual among membrane lipids because it can undergo a reversible phosphorylation at a number of sites, or we can say it undergoes phosphorylation at number of multiple sites on the inositol head to generate a variety of phosphorylated PIs, right? Lipid which are called as phosphoinositides. In a cell signaling event, the enzyme that directly converts your PI, that is 4,5P2, that is PIP2, into your PIP3 is which of the following? You have to find out in the cell signaling event, which enzyme helps in the conversion of PIP2 into PIP3. See, in this question, from the first statement till here, even if you avoid, there is no, you can save your time. The question is starting, the main question is starting from the last line, which says, in the cell signaling event, the enzyme which helps in the conversion of your PIP2 into PIP3 is which of the following? First one is PI3 kinase, second is PLC beta, third is P10, then you have first protein kinase. Very simple question. Very good, sweetie. Others also mark the answer. Very simple question. Yes, so the answer for this question is option number 1. So the activation of your PI3K results in the phosphorylation of phosphatidyl inositol PIP2 to generate the inositol 3,4,5 triphosphate, which serves as a secondary messenger, and then it will be helping in the conversion or helping in the activation of the number of downstream signaling pathways, right? Next question: which of the following statements about the distribution of the chromosome within the interphase nucleus of a mammalian cell is correct. You need to find out that which statement is correct in terms of the distribution of the chromosome within the interphase nucleus. First, the chromosomes are randomly distributed within the nuclear volume. Second is saying the gene pool chromosome tend to locate towards the nuclear envelope. Third, the larger chromosome tend to locate at the periphery. Fourth, centromeric region of all the chromosome tend to concentrate at the center of the chromosome. Question is asking us about the distribution of the chromosome within the interphase nucleus. When a cell is undergoing the division, or we can call it the cell cycle. So at that phase, you have the interphase and then you have the M phase as well, right? So in the interphase, the cell is preparing to get converted. So in the interphase itself, you have the G1, S and the G2 phase. So question is asking that during this interphase, your distribution of the chromosome is in what format? This is what the question is. Try the question. First is saying that there is random distribution. Second is saying that the gene poor chromosome are located towards the periphery. They are located towards the nuclear envelope. Next is saying the larger chromosomes tend to locate towards the nuclear periphery. Third is saying the larger chromosome are located towards the periphery. And the fourth one is saying that they are concentrated at the center. Very good. Everyone mark the answer. Distribution of the chromosome. Everyone participate in the class. Yes, the answer is option number 2. That is, we need to find out the correct statement. The gene poor chromosome tend to locate towards the nuclear envelope. This is the correct statement. So the gene poor chromosome tend to locate towards the nuclear envelope. Simple and easy question, right? Remember about this. Simple, basic and easy question. Next question. This is from your cell adhesion molecules, which says column X represents the type of junctions and column Y represents the protein that are associated with these junctions, right? So which of the option is correct between X and Y? Very easy. You have anchoring junction, occluding junction, channel forming junctions, and the signal relaying junction. And on the Y column, you have Claudens, delta notch, desmoglein, and connexin. Try correlating for this and you'll be easily able to get the answer for the same. Try it out. Very simple question. Try the question, everyone. Okay. So you have anchoring junctions, clouding junctions, anchoring junctions, clouding junction, channel forming junction, and your signal relaying junction. So very simple one is the channel forming one, right? So the channel forming should be marked with your connexins. So C gets marked with the fourth one. You can eliminate option number D very easily, right? Then you have the signal relaying junction, which can easily be marked with your delta notch, right? Because in the delta notch pathway, we have the signal relaying junctions, the delta and the notch being there. So D gets marked with do.
C gets marked with the fourth one. Then you have the desmoglein. So anchoring junction gets marked with the desmoglein and the occluding junction gets marked with your Claudins. So very, very simple question. One is the correct answer for this one, right? Basic, simple and easy question. There is nothing to even think about in this question. Very, very simple and basic. Next question. Which cell cycle phase is typically the shortest in the mammalian cells? Question is, which cell cycle phase is typically the shortest in the mammalian cell? You have G0, G1, G2 and the mitosis. Very simple again, basic and straightforward question. You have your examination in just 2 weeks. So you should be very much thorough with such simple question. You should have very good knowledge about these questions. Question is, which of the phase or which cell cycle phase is typically the shortest in the mammalian cells? The G0, G1, S and the mitosis. Everyone participate in the class. Very good, very good. Very simple question. G0, G1 and G2. Like G1 and G2 belong to your interphase. G0 is the quiescent stage. If the cell exit out the cell cycle, so it enters into the G0 phase, and mitosis is your main division phase in which the cell is actually dividing in, and as a result the mitosis will be there. So with cell cycle, the cell will be actually dividing, and hence the mitosis is the shortest phase of all time, right? So remember about this particular thing. Mitosis is a phase of cell cycle which helps in dividing the nucleus into its identical nuclei. It involves a number of stages: prophase, metaphase, anaphase, telophase, etc. Next, porins, which normally are present on the outer membrane or the outer mitochondrial membranes. They reach their destination by which of the following? First one is direct synthesis of the porin by on the mitochondria membrane by the mitochondrial protein synthesis machinery. Second, synthesis on the ER and transport via vesicle to the mitochondria. Third, synthesis in the cytosol, import by the TOM and insertion from the intermitochondrial membrane space. Fourth, synthesis in the cytosol, import by TIM complex and insertion in the membrane. Very simple question. Try it out, everyone. Try the question. Very good, Niharika. Others also try it. Okay, let's see. Yes. So porins, which are normally present on your outer membrane, outer mitochondrial membrane, they reach their destination by synthesis in the cytosol, import by TOM complex, and insertion from the intermitochondrial membrane space. So C is the correct answer. Like it's a factual question, we can say. You should know the entire scenario for marking such an answer. Next question. Which of the following is involved in pinching off of the neck of invaginating coated pits to form endocytotic vesicles at a presynaptic terminal? Which of the following is involved in the pinching off of the neck of invaginating coated pits to form endocytotic vesicle at the presynaptic terminal? Synaptogenin, AP2, clathrine and dynemin. Dynemin. Try the question, everyone. Very simple question. Very correct, very correct. So, dynamin is the correct answer. So, which of the following is involved in pinching off? It's a very static and a very straightforward question asked. If you have learned about the endocytosis mechanism, you can easily mark the answer for this question. So, which of the following is involved in pinching off of the neck of the invaginating coated pits to form the endocytotic vesicle at the presynaptic vesicle terminal? Answer for this is dynamin. Very simple question. Bhageshri, answer clathrine is the coat. It is not asking about the coat. It is asking you about the protein that helps in pinching off this clathrine coated vesicle. Next question, give it a try to this. Following are the marker enzymes that would be used to identify the correct subcellular fractions. Following are some of the marker enzymes that would be used to correctly identify the subcellular fraction. So, which of the following option correctly pair the enzymes with the subcellular fraction? So, you have the enzymes and the fraction. So, you have lactate dehydrogenase, for acid phosphatase, glucose 6 phosphatase, and catalase, and the various organelles like lysosome, microsome, cytosol, and peroxisome. Try the question, everyone. Simple and easy. Okay, let's see. So you have lactate dehydrogenase, acid phosphatase, glucose 6 phosphate, and catalase. Catalase is directly like related to your peroxisome. So you can eliminate option number second and fourth easily. D gets marked with the fourth one. Then you have lactate dehydrogenase. One of the option is given to you as lysosome, like either one or three, either lysosome or cytosol. Lactate dehydrogenase will be marked with your cytosol. Acid phosphatase will be with lysosome because acid phosphatase has functionality in lysosome and glucose 6 phosphate in your microsomes, right? So 3 is the correct answer. This one is again a factual question. You have to remember the static factual informations for marking the correct answers. Very good. All three of you got the answer correct. Next question, give it a try to. Let's do this question. So you have some of the protein modifications and their functionalities. Okay, today the questions that we are discussing, all of them are from December 2023, like the latest year paper question. So try the question, everyone. The table below represents some of the protein modifications in the column X and their function in the column Y. Which of the following option represents all the correct matches between column X and column Y? Try the question, everyone. Mannose 6 phosphate, try to correlate it to the most logical terminology. Try correlating mannose 6 phosphate to the most like relevant terminology. Mannose 6 phosphate is somewhere related to the lysosomal targeting because for targeting any protein towards lysosome, you require mannose 6 phosphate. You can eliminate option number D from here. Answer is either A, either B, or C. Then you have glycosyl phosphatidyl inositol. So first should be marked with your aromatization is protein degradation, sumolization is protein sorting, and phosphatidyl inositol is membrane anchoring. So second should be the correct answer and not the fourth one. You can easily eliminate the fourth one. So second is the correct answer for this particular question. A gets marked with the protein degradation, sumolization, protein folding, glycosyl phosphatidyl inositol with your membrane anchoring, because it will help in the anchorage of both the things. And then you have mannose 6 phosphate for lysosomal target. Yes, very good. Second is the correct answer. Next question. Which of the following statements about the biological membranes is incorrect? We need to find out which of the statement about the biological membrane is incorrect. First one, the lipid molecules can diffuse freely in the plane of the bilayer. Second one, the ratio of the size of the head group to the fatty acyl chain contributes to the curvature property. Then, individual lipid molecules rapidly rotate along their long axis, and phospholipids freely diffuse from one leaflet to the other one, right? So remember about this particular thing, everyone. Which of the following statement about the biological membranes is incorrect? They can diffuse easily, freely. Then ratio of the size of head group to the fatty acyl chain of the phospholipids contribute to the curvature. Then small modification in the ubiquity. Symbolization. B gets marked with the first one. Wait, I think I made a mistake over here. Yes, polysomalization is related to the protein degradation. Yeah, it is small ubiquitin like modifiers. They are helpful in the degradation. See, regulating various aspects, correct. But the degradation is majorly via ubiquitination. These are modifiers. They are not just for the degradation. Like, let me just cross check for the palmatization. Membrane association. Palmatization is membrane association. Okay, wait. First gets marked with second, then D gets marked with palmatization is for sure membrane anchoring. Let's just cross check this. Really sorry. Just a minute. Palmatization, that is A, should be marked with the anchorage because it is helpful in the anchorage. First with second, you can eliminate this and this. Then D with 3, that is mannose 6 phosphate with 3. Then protein folding is there and protein degradation is there. See over here, polysomalization, if it gets marked with the protein folding, this gets marked with the degradation. But no, that's also not correct. I guess there's a problem with options. Glycosyl phosphatidyl inositol. Just a second. It is for the protein folding because it is having linkers. Yeah, it is helping out in the linking as well. So it can be helpful in anchoring of a membrane to this. So they are marking C with second also. One with anchoring. D cannot be the answer. Fourth is not the answer. Just a second. Some problem. No, I've added a wrong information over here. Incorrect. Just give me a second. Really sorry. Just cross check. Palmatization. Position should be marked with the number. If one gets formalization gets okay, B gets marked with the second one as according to many students, but it can be marked with the fourth one as well. It's okay. D with this with degradation, but no. I think the answer should be option number B only. Niharika, I think the answer should be B itself. This getting marked with a membrane anchoring. C with second is correct for sure because inositol anchoring. C with second. Polysomalization, some of you are saying as 1, calling it 4. Mano 6 phosphate is this. Let me cross check it properly and I'll get back to it. It should be marked with membrane. Palmatization, okay, according to you. Anchoring, okay, according to you. First, I should be marked with this. Membrane anchoring, linking. But glycosyl phosphatidyl inositol should also be marked with membrane anchoring itself, right? Because it is helping out in anchorage. It also helps in anchoring only. It helps in anchoring a membrane to a protein. So C should also be getting marked with 2 according to the correct answer.
Should also be getting marked with that itself, right?
So I'll cross check this. According to me, the answer should be like, there's a confusion in the options. Palmatization if membrane anchoring, phosphatidyl inositol also with the membrane anchoring. Degradation protein folding. So if you are marking C with fourth, A with second, first, okay, 1 can also be the answer, right? According to what Niharika is saying, A can also be the answer. I'll cross verify it tomorrow and I'll like get back to this question in the next class. Like tomorrow also we'll have a live session. So I'll take up this particular question tomorrow only. Okay, let me check what's the wrong thing that I'm doing over here. I'll cross verify it. According to Niharika, the answer is option number 1. Palmatization, membrane anchoring, polysomalization with protein degradation, this with anchoring and this with lysosomal folding. I'll check it. Okay. Really sorry for this. I think answer should be option 2, but one of you saying the answer is option 1. I'll cross verify it and I'll let you know. Okay. Because B. Anyway, I'll check it tomorrow and I'll get it done. Okay. No worries. Let's meet in the next class. Okay. In addition, we are starting off with our full course for the December 2024 examination very soon. If any one of you who's interested in joining up the batch for the December course, you can download our application and get yourself enrolled in the same. All the links are in the description box. Okay. Let's meet in the next class tomorrow. Bye everyone. Take care. And also do not forget to hit the subscribe button. What means